Okay. A couple of opening remarks. Letting speak somebody else in my for me, and that's Jim Hurley. When we had this key publication on the ESCRT system, these are these spirals that your cells use all the time when you have cell division, but also these molecules are hijacked by, for example, HIV and certain viruses when they use to do abscission from cells. So these are protein structures that work like mechanical spirals that load. We showed that they are loading actually, and so they pinch off membranes. And when we published this work, Jim Hurley wrote a commentary and said, high-speed atomic force microscopy is the main enabling technology for these new advances. These devices have the potential to revolutionize our understanding of protein self-assembly on membranes by providing single protein structure resolution in real time. And obviously it's my objective to convince all of you of this revolutionary character. And I have also, in my last slide is a call that I have open postdoc positions, especially for physicists that like to develop new things. So that's the last slide. I will not talk over that. So how do we make AFM better, right? So not one thing is tips, right? The sharper the tip, the better the image. And we actually use for high-speed AFM these very short, ultra short cantilevers that are only about 8 micrometers long, so about 20 times longer than shorter than conventional AFM tips. And so they have a resonance frequency of about 600 kilohertz in liquid while still being relatively soft. And so this is one of the major tricks to make high-speed AFM about between 100 to 1,000 times faster than conventional AFMs. And then what we do is we have an SCM in our lab, and we put the AFM tip into the SCM, and we consciously contaminate our SCM. And then we zoom with the SCM at the end of the tip and switch off the imaging mode of the SCM so that the electron beam burns down exactly at the end of the tip. And then basically you ionize these molecules that you put into the electron column, and you grow over about a minute a very long, beautiful needle that has a high aspect ratio and sharpness. And we use then also plasma cleaning to sharpen this tip. So when you use the AFM again, you know, you contaminate your tip, so you can plasma clean it and you sharpen it again. It gets shorter, shorter, and shorter when you do this. So you can do this like 5 to 10 times. You have to regrow your needle. It's obviously time consuming, but in the end, failed AFM experiment is more time consuming. Okay, so what we also did, and this is obviously a homage to early works in Hermann Gaub's group, is our work on titan force spectroscopy when we developed high-speed force spectroscopy. And the key thing was that classical AFM experiments measured with a couple of 100 nanometers of per second pulling the titan apart, that this key experiment by Matthias Rief in the mid-90s. And at the same time, you may know that Klaus Schulten then started to pull the titan apart in the computer. And obviously this has been done because in computers you can only, in the best people could simulate a couple of nanoseconds to unfold, and these proteins, when they unfold, are 25 nanometers long. So they had to pull tremendously fast to stay in computational timescales. And basically what we did is then modify our high-speed AFM with the short cantilevers with these large response times, and we reached 4 millimeters per second pulling speed. Obviously now the temporal resolution gets limiting, but the key is this data point here that overlaps. That was the first experiment that overlapped experimental unfolding of titan with molecular dynamics simulations. And so that is one of the key things, but I'm not going to talk about force spectroscopy today. So we developed further things like environmental control for high-speed AFM. And here you see the high-speed AFM setup. We implement a pumping system or some microfluidics. And so we can change the environmental conditions while watching the single molecules. And this is annexin. This is a molecule that adsorbs on a membrane, negatively charged membrane in the presence of calcium. And then while I'm talking, we are titrating EDTA into the solution. So EDTA, we pull out the calcium, and then you have these single molecules going away from the membrane. Another example is here is the toxin. It's a toxin from the earthworm. That's the molecule why you shouldn't eat earthworms. So the brighter molecules are the so-called pre-pore state. In the pre-pore state now, 95% of the molecules are in the pre-pore state, and they sit on top of the membrane. So there's a supported lipid bilayer underneath. And then when you acidify, then you can see they pop into the membrane. So now you can see, if you, for example, observe single molecules, you will see as we acidify, then they go into the membrane, and you see these darker spots. That's the pore state of the molecule. And we can do this also by using a UV laser that's injected into the high-speed AFM to uncage caged compounds. Like nowadays, people can create caged protons, caged calcium, caged ATP, and so on. This allows you to play to some extent with the molecules. Environmental control light, obviously. Key experiment, bacteriorhodopsin. Everybody knows bacteriorhodopsin. This is the bacteriorhodopsin D96N mutant. Is a slow mutant, and so here you can see the movie. So we start here in the dark state. This is the correlation average, and then when we switch on the green light, you can see this dramatic conformational change as the X-ray structures show. In the dark state, light state, the F loop moves out, so you can monitor this conformational change. Since this is the slow mutant, and I come back to that, it has very long tau. So this is just 2 frames, 72, 73, and then you can see the single protomers falling down. Obviously, in the correlation average, it looks like they all come down back to the ground state somehow in a concerted way. That's not the thing. Each molecule comes stochastically back at a given time point, as shown here. Temperature control next. So we developed a system where we sputter a tungsten film underneath the glass slide of the high-speed AFM fluid cell, and we use it as a resistor, so we can apply a voltage on the tungsten coat and heat up the fluid in the fluid cell. And this is showcased here on a lipid bilayer. This is called DMPC, and DMPC is an easy target because it has these so-called ripple phases, coexistence of liquid, crystalline, and fluid phases at room temperature. So you see here the stripes. Now it melts here on top. Here it gets smooth. And then when we cool it down, then these ripples come back concentrically. We believe it's like when you take the cake out of the oven, it starts to cool down from the side. And in these experiments, we try with a feedback loop on the temperature to place ourselves on the transition temperature of the molecule, of the lipid bilayer, and kind of play with it as it goes back and forth from liquid to this ripple phase transitions. Next, obviously, force. Force to control force is actually not so easy in a force microscope. And because it relies on 3 parameters. That's the free amplitude, the set point amplitude, and the measured amplitude. And just to give you an idea, so obviously once you start imaging in an AFM, you're all permanently feeding back on the set point amplitude. So set point amplitude is no problem, right? So it measures the amplitude of oscillation of the cantilever. The amplitude detector does that, gives the feedback, and that drives CPSO to always keep the set point amplitude constant. But unfortunately, after a while you're into the experiment, you start to forget what A3 is because you have fluctuations, temperature changes, and so on, and evaporation. So A3 actually changes quite significantly. So we developed such a free amplitude and force controller. And the trick is that every time, so this is the scanning signal that we send to the X piezo. The X piezo is left, right, left, right, left, right. So this is left, right, right, left, right, and so on. Every time that this pulls around, going from right to left, so like when you read a book, you read the line from left to right. Our images and movies are always constructed from the trace scan line. So every time we go backwards, this electronic circuit sends a fake signal to the PID and says, hey, there's an obstacle. So what the PID does, it pulls back, it sends a signal to the C piezo, it pulls back. And what happens then is that during this very short period, while we're going back, it's like when you take your eyes off of the book, right? You refocus, and we measure briefly what the free amplitude is. So in this experiment, we set the free amplitude to 1 nanometer and the set point amplitude to 9.2 angstrom. And then we have basically the second feedback that runs and adjusts the excitation. So we're controlling a free and a set, and this equation is an approximation of the average force applied. And so basically when we are switching on our circuit, then we can control a free over a set constantly. If you switch it off, somebody walks into the room, your A3 is going to change and so on. So this is a very important development to actually to have like a real force control over in AFM. Now about A measured, obviously your interest is to measure as quickly as possible what the cantilever actually sees. And the conventional method that is done in oscillation mode AFMs is to do a Fourier decomposition. So you just analyze the oscillation of your cantilever by fitting make Fourier decomposition and take the amplitude of the first component. That gives you obviously a delay of 360 degrees because you get a single value at the end of every oscillation. So now we reasoned that we can simply use this trigonometric consideration and shift the oscillation of the cantilever by 90 degrees and do this operation to take the square root of the oscillation time plus the square root of the oscillation shifted by 90 degrees and the square and then the square root out of it, and we get A out. And obviously this should have just a 90 degrees phase delay. And this is shown here. This is actually really working. And this is our first, let's say, product where we put everything together. This is a real oscillation trace of a real cantilever. So you can see here, for example, it's a kind of a smaller amplitude followed by a larger amplitude. And our new amplitude detector follows that detail, while this yellow-greenish trace here is the Fourier method, and it comes late. It comes an oscillation or two oscillations late, analyzing this and counter works there actually. So these developments have allowed us to, for example, very nicely to study the piezo channel. And for those of you that seen that Nobel Prize in 2021 to Artem Padaputian, the piezo channels essentially allow you to sense your fingertips, allow you to stand upright, control your blood pressure. Everything in your body is done by this molecule. And we have then in this movie, we have these piezo channels with the characteristic extracellular domain. It's a strange membrane protein that sits actually in such a ball shape, bends the membrane, and actually that's the trick that it does when you apply force, and we apply then the force by varying this ratio of A set over A3, which is very intuitive. The harder you, so we keep obviously A3 constant with our A3 stabilizer, and we diminish then the ratio of A set over A3, which is like when you hammer a nail into the wall. and then we can derive the force that we apply as a function of time in this movie. And we see then how the piezo channel expands in the membrane under force, and we can get the spring constant out of the piezo channel in the membrane. So breaking resolution limitations obviously is a big one for us, because AFM is a little bit what was called blobology. And so this is has been inspired obviously by super resolution optical microscopies. So the idea is that due to the finite tip size, you convolute the sample, right? It's fundamentally limited by the tip size of the AFM. When you apply a force, you have an increased contact area between tip and sample. In biological samples that are soft, this is an increased problem. Then proteins are flexible structures, so they kind of fluctuate around, so that will average in addition out. However, we know from very early, from the earliest AFM data, that the XY position stability of an AFM is sub-angstrom. Obviously, you can image atoms on solid surfaces, and the topographic resolution might even be better, as we know from atomic imaging, but also, for example, from resolving phase separations in lipid bilayers due to the near field effect. Obviously, the C resolution is tremendously high in an AFM. So we figured that maybe we can take advantage of the flexibility of proteins. And the idea is the following, like in photoactivated localization microscopy. If you have two fluorophores that are too close together, people have found ways to either switch one or the other one on and off, and then basically fit these point spread functions. And then the resolution, when you merge these fits, the resolution of your super resolution optical image is not diffraction limited anymore, but it's much more defined by how well you can define where the origin of this point spread function sits. And we made the same rationale. An AFM tip will convolute, let's say, the atomic details on the protein surface. We cannot resolve them. However, there's fluctuations, and from time to time, one atom fluctuates up and another one fluctuates up. And because of the high sensitivity of the near field effect by an AFM, we can detect this, and then merge these localizations and get high super resolution AFM maps out. And so the rationale is shown here. Here simulated traces. So if you have these two features, you have these traces, simulated traces. If you calculate an average, you have typical AFM blobology. However, when you just take the peak positions, you go into every trace, you fit the peak positions, you extract the height of every peak position and the probability of peaking, you get a super resolution map out that has in every pixel the real height encoded, and it has also the probability encoded. And this is shown, for example, here, pixel number 2, and these two images are exactly the same. However, in this case, the left pixel is a little bit higher than the right, and here the right pixel is a little bit higher than the left. So the super resolution maxima shifts in these two pixels. You can merge it, break the resolution limitations and the tip convolution. We needed to develop a new AFM color scale. Typically in an AFM color scale, it's just from black to white encoded the heights. So obviously, if you have a high but flexible element, then it would be like half the probability to pick it if you just translate it into one such thing. Color scale, this would give the same result, something that's half the height but very stiff. So we developed this new 2D color scale where we are encoding in a false color scale the height. So the ratio of the green over the red channel gives you a linear progression to height. Obviously, you probably know that if you have 3 pixels, one is to RGB to 1060, 150, 140, 40, 100, or 70, 20, 50, it's the same color. but it's not the same intensity, right? So it's very clear that in the ratio of green over red, you can encode something, the height here, and in the intensity of the pixel, you can encode the probability of picking. So you have the full information. And now this is showcased here on an old AFM data set. That's an aquaporin that I took actually in Basel during my thesis. These are the detections. And when this starts again, you will see how these detections fill up this probability space here. And this is the surface of the X-ray structure. And now this is just the correlation average. And now you can see how we get, if you just average all these molecules together, you get the AFM blobology. And if you go to this localization AFM procedure, you get almost atomic resolution on the surfaces of the protein. So we analyzed this in two ways. We used the Fourier ring correlation function, that is a classical method in electron microscopy, or we just looked in real space by separating the data set in two half maps. So the two half maps contain no particle in common, and you calculate two independent averages, and then you compare them either in frequency space or in real space. And then we could prove here that we indeed resolve features on the surfaces in the two half maps that have no particle in common, that are 2.6 angstroms apart. So we can also do this on other molecules. This is a CLC transporter where they are not so densely packed. For example, these guys, they move a little bit around, they rotate from time to time a bit. So all you must do is do some classical image processing, like extracting molecules, center them and align them. And then once you have a particle stack, you can run your localization AFM method. Here again, particles, detections, the localization AFM map, the X-ray structure, stunning correlations that you get. And in this case, we could image, since it's a proton-activated transporter, we could analyze the conformational changes of this transporter as a function of pH. Good, now we propose that we can use this method in two different ways. Either we can image, let's say you have the possibility to get a high-speed AFM movie of, let's say, 50 molecules. You could calculate a super resolution map of these 50 molecules and then like run a running window over your movie and get high resolution frames of each individual frame, and then have a time resolved localization AFM map. Or you could do it the other way around, okay, and that means that you look several times at the single molecule and actually calculate a quite high resolution structure of an individual molecule. And I think this has a lot of promise, because let's assume we have this problem that the molecules are stochastically sometimes not working for 30 seconds, right? Although they should transport. So one of the hypotheses to explain this is that sometimes in molecules a loop unfolds. Sometimes you have a little element in a protein that unfolds. So this method, then you could calculate a rather high resolution image of a single molecule as it traverses during its life, and possibly detect such events of unfolding or partial state dependence as a function of time. Okay, breaking speed limitations of AFM. Obviously, high-speed AFM did that already by a factor of 100 to a factor of 1,000, as shown here on the slow bacteriorhodopsin mutant. But now, obviously, that still is, for biological processes, not good enough. So we reasoned, if let's say we take here this movie at 2 frames per second, and every image is about 500 lines per second per frame, so we have about 1,000 lines per second. So now we just basically unplug the Y scanner, the slow scan axis, and then we are getting 1,000 times the same scan line and stack that in a chymograph. And now we're looking at wild type bacteriorhodopsin. And now this is in the dark, and then we will see as we switch on the green light, this line becomes green. Then you will see how the molecules start to fire. now you can see the molecules start to fire. So that's the EF loop moving in and out. And obviously you have now a millisecond temporal resolution. You can resolve the conformational cycle of wild-type bacteriorhodopsin. We can even drive it further, and this has been also inspired by FCS, is basically to say, okay, some processes are so fast, not even the scan line is good enough. Just let's not use the AFM as an imaging tool anymore. Just park the tip at a specific position. So we are targeting a molecule, basically switch off the scanners and still have a feedback, but now we have about 500,000 oscillations taps per second, about 10 microsecond temporal resolution, and then you can see the fluctuations of the molecule that either you can detect diffusion or conformational changes under the tip, like that. So how do we do this? And now I'll give you two examples, bacteriorhodopsin. So as a quick reminder, bacteriorhodopsin, you have, this is extremely well studied. It's maybe the best studied membrane protein by obviously the whole history of German biophysics, starting with Dieter Oesterheld. And you have the retinal in the middle of the bacteriorhodopsin that absorbs the light, and then you have this cis-trans conformational change, which leads to proton pumping, where a proton is expelled on the extracellular side. Then you need to reprotonate, and that's why this mutant D96N is so slow, because this D is important in the reprotonation pathway. So then it opens that gate on the intracellular side, lets a proton in to reprotonate the Schiff base. So that's more or less the trick in a nutshell. Now, obviously, we have 50 years of spectroscopic studies about it. And the important point here to take home is this time constant of 12.1 milliseconds. Here, this is when the pyranine monitors this process, the reprotonation. So they have this complicated systems. Obviously, they can look spectroscopically at the retinal, and they have two fluorophores, the pyranine and the fluorescein, one attached to the BR and one in solution to monitor protons coming in and out. And then you have obviously time-resolved X-ray crystallography that gave you the structures, how the EF loop moves out and so on. So based on this 12.1 milliseconds, these people think that it works at about 100 per second. In X-ray crystallography, it's obviously very slow because it's in a 3D crystal. So they say that the structure changes over 200 millisecond time, complete the photocycle. So that would be about 5 per second. So we use then this high-speed AFM method. Don't talk about the mutant anymore. You understood how we do that in imaging mode. So then we said, okay, let's try to watch the wild type. And as I just described before, obviously in imaging mode, even at 5 or 10 images per second, not good enough. All you can do is get some noise. This channel is just a standard deviation between frame N and frame N+1. So you can see some noise when we switch the light on, but we couldn't resolve the conformational changes of wild type. And that's, as I said, when it came on, this chymograph strategy to get millisecond temporal resolution, we get very nice high time traces of these molecules. We can derive then the state dwell times. So then, based on this, we started to play with light. So we started to use low light, high light, and then dark to convince ourselves we're really watching these molecules do their stuff. And as you can see already here, I guess, that the activated state dwell is constant, but obviously when you switch off, give more power in the light, the probability that the photon is captured increases, so the inactivated state dwell time gets shorter and shorter with light. And so we did this in much more detail here, where we are cranking up the light. So this green triangle illustrates the thing. And as you can see, again, the activated state dwell time stays constant, but the inactivated state dwell time changes, get shorter and shorter. And so the activated state dwell time is always about 13 milliseconds, but the inactivated state dwell time gets shorter and shorter, but it tables out at about 40 milliseconds. And that's kind of the surprising thing. What we also did is to measure how quickly does the conformational change come if we are using now pulsed lasers. So we give a laser pulse and then ask the question, how quickly after, if you zoom into these events here, how quickly after the laser pulse came, the loop moves out. And this is in average about 2.9 milliseconds. I don't go into the details of pH dependence here. So basically then we came up with this thing that if a photon hits about 2.9 milliseconds later, we reach the open state, and the open state is 13.2 milliseconds. And as I told you, Densher measured 30 years ago, these 12.1 milliseconds, and they thought, well, that's the turnover rate. There's not much of a reason why that shouldn't, because they used ensemble measurements. However, if you look at the single molecule, there was this surprisingly long dead time that the molecule cannot activate again of about 40 milliseconds. And so we looked into the literature, and in Werner Kühlbrand's lab, Janet Wang has an interesting finding. She says, based on 2D crystallography, she analyzed when she activated, light activated PR, what's the density of this movement of the loop outwards. And she says in the abstract, limiting the, so to change conformation simultaneously, limiting the occupancy of the state in the membrane to 33%. So since the bacteriorhodopsin forms these trimers, she found actually that the activated state, the helix out, only is a third of the density that you would expect, obviously in averaged. So we propose that this is one protomer firing, and now it's silent, but it's only silent because mainly now the other two can work. And so there is this cooperativity. So the maximum reaction cycle that bacteriorhodopsin can run in a native membrane is 20%. That's the take-home message. Last part, oh yeah, I still have a bit. No, yeah, I have a bit of that. 2 minutes. Membrane-mediated interactions, membrane-protein interactions. This is unpublished. So we were wondering about the following, right? In a native membrane, membrane proteins, now we know from fluorescence microscopy, biochemical observations, structural studies, we talk about lipid protein rafts and nanodomains and so on, supercomplexes. So there is a reason that membranes are organized. They are not unorganized. And as soon as you accept that certain interactions are energies are higher than KBT, then you accept that there is some sort of order in a membrane. However, there is not a machine known in biology that takes a membrane protein and says, you have to go over there, you have to go over there, you have to partner with this molecule and so on. So there must be physical grounds to that. And obviously we are not the first ones to invent that, but that's how membrane proteins sense each other through the membrane. So we designed this very bottom-up experiment. We create these 2D crystals of the aquaporin, that is our working horse in the lab. And then we mix this with empty vesicles that we adsorb onto the membrane. These empty vesicles will fuse with these patches, and then the proteins diffuse into the membrane, and we can measure the association-dissociation dwell times. Let me show you the movie, you understand it immediately. So here is the high-speed AFM movie, and then after a while we add the empty vesicles. You will see it starts here, where the empty vesicle comes in. Now, boom, more empty membrane comes in, and in the end it covers the entire surface. And now the aquaporins start to diffuse into that empty space. You can see everything. And after about 500 seconds, we reach equilibrium where you can see that the entire membrane space is now full of diffusing molecules, okay? You can distinguish it because the membrane space of molecules diffuse is notably higher than the empty membrane in the beginning, right? So we know that. So, and then super talented graduate student look at these movies. This is also in our lab by our standards, just outstanding data. The detail, the resolution, the clarity of these single molecules hopping in and out is just really very beautiful. So basically then obviously what he's done the same experiments in 4 different lipids. And consciously we keep the head group constant, everything constant, the unsaturation constant. The only thing we change is the length of the hydrophobic part of the lipid, because of membrane interactions are guided by the cost of bending and deforming the membrane around membrane proteins, which is called a hydrophobic mismatch. So obviously a membrane has its natural thickness. And then if you are thinner as a membrane protein, you thin the membrane. If you're thicker than the membrane, you thick the membrane. So there's elastic costs. So there's compression costs and bending costs to a membrane. And this has been, by membranealistic theories, has been relatively well described. So now the idea is that if these are 2 membrane proteins, and the membrane is thicker, if you move these 2 guys together, so these are the thickest membrane, the thinnest membrane, if you move these guys together, at some point, obviously the cost of this bending and compression around each molecule disappears. So there is an energy gain, a free energy gain in putting these things together. At least this is a one-dimensional representation. Obviously in 2D, the whole thing is a little bit more difficult. So we do this experiment that I've shown you in all these different lipids, and we measure the association dwell times of each individual, in each of the molecules in each lipid. What we also have to consider in this system is that some molecules, as you can see, it's kind of a checkerboard. Some of the molecules have 2 neighbors, those that go into a corner, and some have only 1 neighbor, like this guy here. And so the 1 neighbor ones have a very short association dwell time, while the 2 bond events have longer association dwell times. Now we can use this strategy of parking the cantilever. As you can see here in the bilayer, we only see these stripes. Don't be mistaken, these stripes are all diffusing aquaporins that we obviously don't see in imaging mode. But now we can park the cantilever into these areas here, and then we watch this high time trace. We used exactly the same equations as Carsten has presented before to define the diffusion coefficient of these molecules under the tip and the 2D concentration. And now you can see that, for example, for the diffusion coefficient, the interesting thing is that molecules diffuse slower in membranes that have a higher match. So this has been predicted too, this deviation from the Safman-Delbruck behavior of diffusion, because a membrane in a mismatched membrane drags along a larger patch of membrane with itself. And then we have these dwell times, how they react depending on the interactions as a function of the lipid mismatch. So we can calculate the energies out of these interactions. And to finalize it up, we also use a mutant where we have here, where we mutate this phenylalanine, this tryptophan to an alanine, that allows then the lipids to enter more between the protomers. And then we are even able to see non-tetrameric aquaporins, as you can convince yourself in these individual observations. So we can see tetramers going to trimers and dimers and so on. The statistics are not fantastic of this analysis, but we can get a guess about the energies that hold the protomers together. So summarizing this up, using from conventional AFM, that was a static method. We use high-speed AFM, we reach sub-second. On very small areas, we can maybe get to 100 frames per second. but really important kinetic information on fast biological events we get at this point by using this trick, disabling one dimension of scanning acquisition. Then we do this height spectroscopy. We reach 10 microsecond temporal resolution. The openness of the system allows us to change pH, ions, ligands, light, temperature, and force. And finally, we also succeeded to break the resolution limitations recently. And then I would like to advertise the people who did the work. So Yining is the one who did this last work on the aqua points. Alma did the work on bacteriorhodopsin. George Heath is now a faculty in Leeds who did the work on the super resolution optical microscopy. And a lot of the development work is done by Tsushi Miyagi, who's a research assistant in my lab. This is Weill Cornell. Here, this is where we work. This is Manhattan, Upper East Side, super cool, good salaries. And this here is called the Roosevelt Island. And you come on this Roosevelt Island over this bridge. There is a funicular, that a cabin car that goes over the river here. And this is actually postdoc housing, subsidized postdoc house. The graduate student housing is right next to the institute. Yeah, so obviously, as I said, it's a medical institute. So for me, it's difficult. It's easy to get biologists. It's difficult for me to get physicists and people who are actually being able to develop new technology. And so if anybody of you is willing to build the next generation high-speed AFM, please send me an email.
Thank you very much. That was definitely great advertising. Speed talk on speed AFM. Questions, please. Beautiful talk. Thank you so much. I have a question about the last part. So you sketched this very roughly how these bilayers form, and there's a microsurface and there's some water and then there's a bilayer. Doesn't this make it softer and less easy to image?
Well, all our experiments are done, all the experiments that I've shown here are done on mica. And everything is done in buffer solution, not in water. So we typically have a physiological pH and about 150 millimolar KCl or NaCl in solution. And yes, since we measure these beautiful diffusion events, right, as you can see, we are very sure that the molecules are not stuck on the mica surface. And this is likely due to maybe 5 angstrom to 1 nanometer buffer layer somewhere between the mica and the sample. That obviously this is just physisorption. There's no chemical interaction between the protein and the surface. That's why they can diffuse. Is that answering your question?
Yeah.
Okay. Yes. Very nice talk and awesome movies. I was wondering, could you show the first slide of the tip localization again?
You mean the one with the aqua points or the method?
The method.
This one?
One later.
This one.
Yes. So you are imaging now objects that are much smaller than your cantilever, right?
The tip, you mean?
Yes, the cantilever tip. Yes.
Mm-hmm.
And
I mean, resolving later.
Resolving later, yes.
Yeah.
And I can see that you still can see where those peaks are, but how can the cantilever know how deep the minima between the peaks are?
That is a good point. So in the end, you think that you fill in with this probability density function the entire image at some point, right? So you're giving, how to say, we in detail, we also give the detection is not only a single pixel or a single XY coordinate, we're having a uncertainty of where, give it a 1.4 angstrom uncertainty where the detection is, and attribute the same height to that, unless we have a real detection that gives you a real value there. I hope it's not that easy to explain, but the idea is that you fill up the space with meaningful information, but obviously in the areas where you have detections, I think we fill up the space rather well. Yeah. Yes. That's what I try to say here. It takes X, Y, and Z, and the Z is the same height, the Z as in the average traces, right? It's not changing the C height. Obviously, it's just much less data because you don't have the convolution. And then you sharpen it somehow with the probability where you have the detections. Does that maybe also answer your question better?
Okay. One more question. We have to shorten the answers, and then
I will
break. Yeah, please.
Thanks a lot for thanks a lot for inspiring talk. A more technical question about VR. Are you using the VR OTG? I mean, are you using on mic also the VR, isn't it? And this membrane molecule usually is in a vesicle. Is the vesicle totally kind of collapse on itself when it's immobilized on the mica, or it kind of forms a monolayer of BR on the surface?
Yeah, BR, I don't have an overview image, but it's rather well imaged by AFM. BR is literally 2D patches. It's not even vesicles. Vesicles typically pop open. BR is sheets. It's very convenient for it.
How do you do that with the detergent cleaning or
No detergent in the case of BR, never. BR never sees detergent.
Oh, this is coffee break discussion. You move on. There was one more question. Yeah. Here.
Thanks. Hi. Thanks for this really nice talk. I wonder, I mean, you have this tip and you have this basically buffer solution contains, like in a physics way, a lot of dirt. And okay, you mentioned that you can clean the tip by plasma etching, but I mean, I would have imagined that the attachment time, so that like the time you can image before the first buffer molecule attached to the tip basically makes imaging difficult is rather short.
Well, to be honest, always a buffer should not contain much dirt. The buffer should be a clean biochemical buffer with just buffer plus ions. Not that
Okay,
should not contain. It's when you make an error of operation, right? If you pierce with the tip, you apply too high force, or sometimes your sample might be dirty. And, you know, if the feedback loop operation is not fast enough, you pick up a molecule. Sometimes there's tricks, AFM people have found tricks to go onto the mica and hit hard with the tip and sometimes smear it off or so. But typically, my people tell me once a tip is bad, I better go and plasma clean it and start it anew, then continue experiment.
All remaining questions, please, in the coffee break. Let's thank Simon again.
Thank you so much.
Spectacular talk.